everyone. My name is Lena Feleke, and I'll be presenting about global health and chronic kidney disease and the potential of gene therapy. So I'm going to begin with a little introduction about myself. So my name is Lena Feleke, and I'm a rising 12th grader at Martin Luther King Junior Magna High School in Nashville, Tennessee, in the United States. And in terms of some of the co-curriculars and extracurriculars that I'm a part of, I'm a student at the School for Science and Math at Vanderbilt. And through the program, I'm also a research intern at the Vanderbilt University Medical Center. And additionally, I am the Tennessee Technology Student Association state president. And whenever I get the chance to, I enjoy volunteering locally. And in terms of my interests and future plans, I hope to pursue a career in global public health and data science. So in regards to what I'll be presenting itself, I'm first going to define some important terms that will set up what I'll be discussing. So global health is an area of study, research, and practice with the aim of improving health and enabling equity for people worldwide. And from the branches of medicine, I'll be focusing on nephrology and its connection to the global health realm. So nephrology is a branch of medicine that focuses on the diagnosis, treatment, and management of kidney diseases and disorders. And furthermore, chronic kidney disease is of high prevalence and a major global health concern within nephrology. And chronic kidney disease is a form of long-term kidney disease involving a gradual loss in kidney function over time that can lead to kidney failure. So as mentioned before, chronic kidney disease is a major global health concern, and this infographic highlights some various statistics and pieces of information that characterize chronic kidney disease and show its presence as a concern in both developing and developed countries. So overall, more than 10% of the global population have chronic kidney disease, and additionally with that, kidney disease affects around 850 million people worldwide. And in terms of health disparities, people of lower socioeconomic status are more likely to develop end-stage kidney disease, and even in high-income countries. And this infographic also lists some of the top symptoms of chronic kidney disease and also some of the causes of chronic kidney disease. Despite its high prevalence and various complications, the only treatment option available for those with chronic kidney disease are kidney transplants and dialysis, which are both ineffective. And there's also a shortage present within the nephrology workforce globally in both developed and developing countries. And the World Health Organization currently has a list of non-communicable diseases that they prioritize. However, chronic kidney disease is not on the list despite it having a higher prevalence than many of those on the list. And also, research and innovation for kidney disease lag behind those for other diseases. So based on this information, better support and interventions are needed in nephrology for chronic kidney disease. So in terms of kidneys, which normally come in pairs, they aid in the removal of waste from the blood. So unfiltered blood goes into the kidney, and then through various processes, it becomes filtered. And the filtered blood ends up exiting the kidney and going back into the bloodstream. And then the remaining waste ends up going through the ureter, and then the urine exits to the bladder. Kidneys further have building blocks called nephrons, in which the various parts of it are shown in this diagram over here. And with these different parts, they aid in the transport of water and other important materials such as glucose, minerals, and amino acids. And up to 30% of chronic kidney disease is attributable to monogenic renal diseases, as shown in this image over here, that occur in the different parts of the nephron. And this basically shows the potential of gene therapy for slowing down chronic kidney disease progression. And my project, which I will discuss next, focuses on cystinuria, which is based in the proximal tubule of the nephron. And so current cystinuria treatments are ineffective, but up to 70% of cases progress to chronic kidney disease, and the average age of onset is 12.5 years old. So this makes it critical to research therapies to treat cystinuria. So now I'll be discussing about the research that I'm working on now that highlights kidney gene therapy and its potential. And so my work is in regards to assessing the effects of transcriptional and post-transcriptional regulatory elements on SLC3A1 transgene expression for type A cystinuria gene therapy. So cystinuria has different types, but the type that I used as a disease model in my project is type A cystinuria. And cystinuria generally is a monogenic kidney disorder that is characterized by kidney stone formation due to the accumulation of the amino acid cysteine in the urinary tract. And type A cystinuria specifically results from mutations in the gene SLC3A1, which encodes for the protein RBAT. And RBAT is important for the reabsorption of cysteine. However, a mutated RBAT protein causes cysteine to build up and then create kidney stones. And the lab that I am in utilizes adeno-associated viruses, or AAVs, to deliver therapeutic genetic material, which is the safest and most effective. And with AAVs, the material getting delivered is surrounded by inverted terminal repeats, or ITRs, on both ends of the sequence that help with replication and packaging. And the first part of my project has the aim of using and evaluating self-complementary AAVs that use double-stranded ITRs with one full-length end and one shorter length end. And previous work within the lab that I'm in has shown enhanced transduction into the kidney with self-complementary AAVs as opposed to single-stranded AAVs, which consist of single-stranded ITRs with full-length ITRs on both ends. And however, due to the fact that self-complementary AAVs have a packaging capacity that is limited to about half that of single-stranded AAVs, there is an aim to create a shorter plasmid construct to package into the AAVs. And the next aim of my project is utilizing micropromoters, specifically MP84, in the plasmid constructs that are made. And so MP84 is derived from the promoters of human insulin, and has only 84 base pairs while being shown to be as strong as or slightly weaker than conventional promoters. So this ties back to the size aspect that I was talking about before. And the third and last aim of my project is inserting various post-transcriptional regulatory elements and observing gene expression of SLC3A1. So one of the elements that I'm looking at right now is woodchuck, or woodchuck hepatitis post-transcriptional regulatory element, which aids in increasing transgene expression from various viral vectors. And it contains about 600 base pairs, which again ties to that size aspect as mentioned before. And then furthermore, I'm also looking into the type of poly A tail used, in which the constructs that are made will have either SV40 or BGH poly A tails, in which SV40 is the shorter one. So to conclude, more support financially and systemically is needed to advance kidney research and to increase the size of the nephrology workforce. And moving in the right direction requires utilizing data science, using genetic tools to find therapeutic targets, developing various clinical trials with diverse demographic participation, and recognizing health disparities and mitigating them through community-engaged work. And finally, in terms of the direction that I'll be proceeding with our project, I'll be designing different plasmid constructs that will contain different combinations of the elements mentioned before, and I will assess mRNA and protein expression of SLC3A1. Here are my references. And I'd like to thank those in the Matthew Wilson Lab at the Vanderbilt University Medical Center and the School for Science and Math at Vanderbilt for the opportunity and support in regards to my research, as well as the conference management team for GHLC for the opportunity to participate and present my research. And thank you everyone for watching and taking the time to learn more about the topic I discussed and the work that I'm doing in relation to it. And here's my contact information if you'd like to ask me any questions or contact me for other specific matters. And thank you so much for watching.